Bismillahirrahmanirrahim. So we are going to have a review of the last lecture where we talked about cell signaling, and we left at notch signaling pathway, which is one of the juxtacrine signaling pathway, where you can see that the ligand delta is produced by one of the cell and the notch receptor is on the other. And this ligand and receptor interaction takes place, direct interaction takes place, in contrast to the paracrine factors. And notch receptor gets cleaved by specific proteases. And the intracellular domain of the, which is also referred to as ICD of notch, intracellular domain of the notch, after cleavage, it comes into the nucleus and it interacts with transcription factors and eventually leads to activation of different genes. It's crucial for a number of tissues and organ development, in particular the nervous system development. But if you take notch pathway and look at its role in pattern formation during early development, you will find it one of the most critical developmental signaling pathway. And in contrast to the paracrine and juxtacrine signaling, we also talked about permissive interactions or permissive signalings, which provide the environment. So a cell has all the constituents to grow and to develop and get specialized, but it's meaningless if it is not in the right environment. And the extracellular matrix provides that source of critical and developmental signal, or the environment. It comprises of the macromolecules in the immediate environment, like collagen is one, which is an extracellular protein. We talked about fibronectin earlier, and in the vicinity of the cell, immediate environment of the cell, this provides anchorage to the cell. If you carefully look here, you have within the cell, you have these actin microfilaments, which gets connected with the fibronectin through intermediate proteins. Also, integrins play crucial role in this anchorage of the cells within a specific environment. You have many glycoproteins, specialized glycoprotein molecules, and fibronectin is one, laminin is other. We talked about these two in the previous lecture. Besides, we have collagen, glycans, and they all together provide this anchorage or cementing in a calcium signaling dependent or calcium dependent manner. The fibronectin is a large protein, as it is mentioned here. It's like 460 kilodalton, and it dimerizes by numerous cell types. And together with these glycoproteins and collagen and proteoglycans, you have this. This matrix is like a cement. It provides stability to two neighboring cells, and then, of course, to the whole tissue. So with that, I hope it gives you a clear idea about how during early development, in particular at the time of pattern formation, um, or even immediately after blastocyst stage, where you have uh, onset of gastrulation, and then eventually in the form of uh, embryo becomes three different layers. These are the cell signaling pathways which are playing crucial role. These are the cell signaling pathways which communicate, which helps communication between two adjacent cells and cells at distance from each other. And this communication leads to then specialized cell types, which then commit to specific cell fates. And then, from few cells, they become different tissues within the same organ. So this is the picture, or this is the fundamental concept which you should keep in your mind as we move on. So today we are going to talk about uh, fly development. Our first real dev bio lecture, and because this will give an idea about uh, use of all these tools and concepts which we were talking about in the previous three lectures. So in the early part of last century, where fly became one of the favorite model organisms uh, for developmental biologists, and a number of tools, genetic tools, were developed. But as we learned about, we had understanding about DNA with the advent of structure of DNA, with our understanding of structure of DNA and the central dogma of molecular biology. One of the fundamental question which was in front of the developmental biologist or geneticist was how pattern formation takes place, how body gets into a specific pattern, whether you are vertebrate, invertebrate. What is the developmental toolkit in the form of different genes which play a role in pattern formation, which leads to a clear body development. For example, pattern formation here is uh, you start your life as fertilized egg in multicellular organisms, and then through a couple of developmental stages, and I'll soon go through them one by one, you see a well-grown organism which has left-right symmetry, which is mirror image of each other. Then you have very clearly defined head region. Eyes are always on the head region. You have, for example, in this organism, you have antennas. You have this head region, then this is the thorax region, and then this one is the abdomen region. And not only these three major segments of the body, but if you look at the abdomen, it's also giving you a segmented picture, or it's segmented. Now, how this develops was a fundamental question, and it was not that they were just interested in understanding the fly development, but always scientists are always curious about learning how we develop as well. We can't do these experiments in humans, so you go and do these experiments in Xenopus, as you saw in earlier lecture. Xenopus had been one of the very useful model systems when it came to study development. In case of fly, the life cycle of fly spans 10 days from fertilized egg all the way to an adult. It takes 10 days. However, most of the development which we will be concerned with, which we will be talking about in these lectures, is actually here in first 3 hours, rather maybe first not first 3 hours, in first 2 days. And the whole body plan and pattern is laid down in first 3 hours or so. What happens after fertilization, when a female lays the egg, the difference between our development and fly development is all the development in flies takes place outside the mother. So mothers lay fertilized egg, and then all the development will take place here in this egg. In our case, that all the development takes place within the mother's womb. Now, within 24 hours, the embryo undergoes mitotic divisions, specific divisions, which we know as cleavage. And beauty is that the fertilized egg, all the cells, so here after fertilization, you have, after fusion of sperm and egg, the one cell divides into two, and then, these two cells go to four. These are very, very specialized cleavage divisions, which only involve nuclear division, because these nuclei, they share the same cytoplasm. There's one big cytoplasm. And these, as a result of cleavage division, you have these rapid as a result of rapid mitotic divisions, you have increase in the number of nuclei within the cytoplasm. And all of them are highly synchronized. All of them, they divide at the same time. Their time of division is same, and this goes up to first 13 nuclear divisions. First 13 nuclear divisions are highly synchronized, and these nuclei, they don't have their own cell membranes. An embryo, when all these nuclei are at the same, they are dividing at the same time and sharing the same cytoplasm, we call this stage syncytial blastoderm. And this syncytial blastoderm actually lasts like till first 3 hours maximum. When these cells, these nuclei eventually start moving towards periphery, they become here, they get organized from center, all nuclei move to the periphery in the embryo. And then eventually what we see that the cells, they start becoming individual cells. And we call them cellular blastoderm. Cellular blastoderm means now these nuclei, they will start getting their own individual cell membranes. They are no more sharing the same cytoplasm. And this coincides with then the large-scale movement in the embryo, which is the gastrulation, and that is followed by first ever sign of segmented body plan. We see segmentation there. We'll talk about all this in detail. Now, after 24 hours, so this was the 24 hours. I think I wrongly said up to here 2 days. That was wrong. First 24 hours results in this segmented body plan. You have already a very well-defined body axis and body pattern is laid down. Cells within these segments, different segments, which I called as parasegments at this particular stage, they already are compartmentalized. They already know what they are going to make in future. After 24 hours, a larva emerges from this egg cell, fertilized egg, which has gone through segmentation. We call this first install larva, which is very small. We usually cannot see with naked eye. Then it goes to the second install larva within 24 hours, which stays another 24 hours. And eventually we see third install larva, which you will see crawling on the walls of tubes. When you eat fruits like guava, you know, the जो सुंडी होती है वो ये third install larva, okay, which you can see crawling. So this stays there for 2 days or a bit more, and then all the tissues here, they get hydrolyzed. So this is basically metamorphosis. You can see 3 very clear, distinct phases of development, altogether different from each other. And this is the larval stage of development. This was the early embryonic development within the fertilized egg. And then eventually you have the pupa. Now, all the tissues here, which we know as imaginal discs, we'll talk about imaginal disc in great detail in later part of lectures. They get hydrolyzed, and then you see very well-made organs, eyes and legs, etc. The pupa is immobile. It gets stuck against the walls of tube in which we rear the flies. And within this sessile structure, you see the development of this fly, which finally hatches after 10 days. Now, a short developmental time and, you know, genetic tools which enables to visualize all the different developmental stages made fly one of the interesting organisms, because the question was how body plan is laid down and how segmented or segmentation in fly takes place. If you look at your own development, you have left-right symmetry, you have dorsal ventral axis of the body, you have very clearly defined head, eyes on the face, and then you have kind of not thorax, but very clearly defined body part, which is away from legs. So flies gave us answers to the early body plan and pattern formation, etc. And what turned out to be the answers we got from flies, they were shockingly similar answers, which to the questions which people were probing in vertebrates. So yeah, this under scanning electron microscope, if you look, this is how the embryo looks like. These are the I forgot the name. On the anterior part of the fly embryo, so there are appendages. There's a specific name. Anyway, I forgot it. This is for gaseous exchange or respiration. And then the embryo, sorry, not the embryo, the oocyte lies here, sperm, and this is the sperm entry side on the anterior side. This is the segmented embryo, you can see clearly, and then this is the pupa. Now, I think I pretty much explained you the early development of fruit fly embryo, but let's again revise a bit. The fly embryo, up to 13 mitotic divisions, is a syncytium, and by syncytium we mean nuclei share the same cytoplasm. The nearly, and these cleavage divisions are very fast. You are like one in every 9 minutes. And as I already explained you, there's no cleavage of the cytoplasm. It remains same cytoplasm, but only nuclear divisions are taking place within the same cytoplasm. After 12 cell divisions, nuclear divisions or cleavage divisions, we have nearly 6,000 nuclei in the syncytium. And by the 9th division, if I draw it here, so you have like all the nuclei here. And by the 9th division, all of them, they start moving towards periphery like this. And the end result is you have nuclei all over against the wall of the embryo, and we have central region which is filled with yolk or fluid, etc. Now, this is the stage which we refer to as, when all the nuclei move to periphery, we refer to as syncytial blastoderm. Syncytial blastoderm is, I mean, this is a term for invertebrate, which is the fruit fly we are talking about, but this is equivalent to blastoderm or blastocyst stage in vertebrate embryos. By the 13th cell division, these cells, they start becoming individual cells or cellularized. These nuclei, they start getting their own cell membrane like this. It's a beautiful developmental stage. I would encourage you to go to Fly Lab, and if you can set up a small cage and look at early developmental stages, just first 2 hours or so, you will see with your eyes these nuclei dividing and then the cellularization. This is actually a beautiful stage where, if I try to draw it here, you have nuclei initially, and then all of a sudden you see like waterfalls, like cell membrane from this side, it start moving downwards, literally, and it's a synchronized movement, everything like a waterfall, and then you can see physically under the microscope that this is happening, and then the cells become individual cells, cellularized. Up to this stage, when all the nuclei are sharing the same cytoplasm, they all are actually, the advantage of syncytium or syncytial blastoderm is, when they share the same cytoplasm, they are sharing all the resources within cytoplasm, different transcription factors. They are exposed to different concentrations of the same transcription factors. The position effect is taking place. We will look at all this in today's lecture. Now, this is now what summary of what we have learned. So after fertilization, and then by 70 minutes, you can imagine if after every 9 minutes, there is one nuclear division, how many cells you will have there. And then finally you see, nuclei moving to the periphery. All the nuclei on the periphery, this is a syncytial blastoderm, still individual nuclei, not individual cells. And by this stage, you see, if you pay attention here, very early on, within first 70 minutes, the most posterior end of the fly embryo that is separated from all the rest of the embryo, and we call this pole plasm. So pole plasm is the place where the future germ cells are going to be made. You see this pole plasm, so this is not part of this cytoplasm. It is somehow protected from rest of the body because the future cells are to undergo meiosis. So these cells are to be protected from mitogenic signals, from mitotic signals. And after 3 hours, you see cells becoming individual cells, or cellularization is taking place. I'll encourage you to go to a web page called Fly Move, and there they have placed these videos. And if you look now here, you can very clearly see 2 nuclei. There's a whole mount embryo. Basic fuchsin is the dye which stains nuclei, DNA, and we can very clearly see. And this is presumably after the first cleavage division. So after fertilization, we have 2 nuclei there. Then this is the beginning of the stage 2. So nuclei are still located within the anterior half of the embryo. You can see here. Then these nuclei slowly start, scattering or spreading throughout the zygote, and these nuclei, they are still within the 20 to 80% of the egg length, not here, not there, etc. And then eventually you see these nuclei slowly moving towards periphery. You can see. Okay. So go to Fly Move and have a look at these stages. You have eventually, this is important stage, you can see the pole cells. And this is the stage we talk about here. By 90 minutes, you will very clearly see this. By 70 minutes, you will see, at some kind of fluid on the most posterior and in the periphery. And these pole cells, they are somehow protected from the all this, and they are whatever these number of cells, they are going to undergo meiosis and produce sperm cells or oocytes. These are just different sections. So for example, this is in the this is a longitudinal section. You have yolk and vitellophages, and then this is these are the cells on the periphery. Imagine this as a whole, a cylinder, and it's a 3-dimensional structure. All its walls will have nuclei against it, and eventually they will be cellularized. If you have the cross section, you can see And the cross section has now these are the somatic nuclei. This is from this particular place. These are the pole cells, very clear here drawn. And this stage, like between, I think, up to nearly 3 years, 3 hours, at 3 hours, which is, I think, the 13th stage 13, you see this cellular blastoderm. And by cellular blastoderm, this is just the stage where few minutes later you will see gastrulation movements. The mesoderm invaginates. We'll soon see how it moves. And from this two-dimensional structure, this is a two-dimensional organization of the cells, just against the walls. These cells will start moving in such a way that this will be no longer a cavity, and you will have very clearly defined endoderm, mesoderm, and ectoderm. So ectoderm, mesoderm, and endodermal cells. So go to Fly Move page and you will see these videos. So in case of here, if you look at our cells, these are vertebrates, and these are the invertebrates. The fly is an invertebrate. After cellular blastoderm, when you have just the onset of the gastrulation and formation of mesoderm, endoderm, and ectoderm, in case of vertebrates, you have this one is the blue one. This one is the ectoderm. Cells from here, they leave place, will look at all this. They start moving inwards. And this is called the primitive streak or early neurulation stage. And as a result of these cell movements, you see very clearly this orange color is the cells which are mesoderm, and this one is the endoderm, the yellow one. Now the cells which are here, they are going to make your gut, liver, and lungs. These blue ones are going to make your skin, nervous system, and other associated tissues. And mesoderm is going to make skeleton and muscle, kidney, heart, and blood. In case of flies or in case of invertebrates, the ectoderm actually goes like this. So you will see an embryo is like this. And ectodermal cells here at the ventral side, they start moving inwards and they make a tube-like structure just like this. This is just a cross section. So this is from the ventral side. Yeah, here this will further make things clear. Now this is the fly embryo, and just after the cellular blastoderm, we see these gastrulation movements, and this one, the red one, which is here, is the mesoderm. The yellow ones are the yellow is the endoderm, and then you have these cells, the red ones, which were initially basically part of the ectoderm. All the cells were basically ectoderm, but this is presumptive mesoderm, I would say, presumptive mesoderm, because these ectodermal cells, their fate is that they will move inwards. And if you carefully look at this picture, this one, there is a depression here. Sorry. There is a depression, and this depression is basically invagination. This is basically inward movement of the cells. They are leaving this place. They are moving one step inwards, and that results in the formation of a tube-like structure. Okay, and this mesoderm goes from anterior, the ventral side, all the way to dorsal side as well. At the same time, what you see, these very clearly defined pole cells. And what happens from the both ends, from the anterior side and from posterior side. So there is a large, because this is the gastrulation movement now, there are large scale movements. If you pay attention, the pole cells from most posterior end, due to movement of cells, they are moving like this. And then from anterior side also like this. So we call this as germ band extension, because these are the germ cells. And what happens now, due to this large scale cellular movement, we will see the connection between these cells and these cells together, and tube-like structure will be formed, which will be the gut. Okay. Now, it doesn't mean that these cells remain there. So this stage is called germ band extension. Germ band extension, which is a result of a very, very specialized cellular division, mitotic division, in which cells just change their connections to the adjacent cells. We will look at these developmental stages in detail in later part of this course. And then after some time, the pole cells come back to their original place, which is called germ band retraction. So I encourage you again and again to go and look at videos. You can type in on YouTube and look for these beautiful videos about germ band extension and retraction. And as a consequence, what happens, you have the endodermal cells making up the gut. Okay, this is yellow one, represents the gut. You have now, so these were the mesodermal cells. So the all this one is the mesoderm, which was the movement of, which was driven from the ectodermal cells moving inwards, and this result in the formation of muscles. These mesodermal cells which invaginate, they later on gives rise to the muscles in the fly. Now, this is also the stage where the founder cells for the early nervous system are defined, and these are called nerve cord cells. These are basically the ectodermal cells in the ventral region, which were here on this side. Now we are talking about the blue cells. Okay, these ones. These ectodermal cells, they form a layer of cells, initially like single layer of cells, and we call them neuroblast. The earliest nervous system cells. And we'll look at this development in detail in our later lectures. These cells leave their position, some of them, they leave their position from this organization, they come within, and then they recruit, or there are other cells as well. There are certain molecules which are involved in this neuroblast formation, cell fate determination, how neuroblasts are determined, like there's a molecule called Miranda, and there's another one, very crucial factors. I forgot the name of the other gene in NAMS and Miranda, and there are three genes which play a role in formation of neuroblast, early neuroblast. So all these developmental changes are happening at the time of cellular blastoderm, when cells become individualized here. Now, immediately after this stage, we have this mesodermal cells moving inwards, endoderm, which is highlighted by germ band extension and germ band retraction. And if you can use, yeah, it was Miranda, Prospero, and I think it was Nums, if I remember the third one. So we have here, by this stage, we have now the equivalent of vertebrate neurulation stage. In case of vertebrates, what we have, which was highlighted here at this particular stage, when the primitive streak, so ectodermal cells, they start moving inwards, and just a streak-like structure we see. After this, we have this notochord and neural crest cells, etc., which is the onset of neurulation. So if the equivalent of that is the movement of ectodermal cells, outer ectodermal cells at the ventral region, and they, if you stain using any of the neural markers within the embryo, with what we see, the earliest nervous system is like this. These cells will be stained like this. This is the earliest nervous system in fly. Okay, very clearly defined cells. Midgut, I told you, is two tube-like invagination from both the posterior side and the anterior side, and they both fuse to form the endoduct. As I always say, it's so precise. Whenever I teach this and whenever I used to look at these early developmental stages, I always wondered how cells moving from two different ends, they meet in the middle, and then they fuse like a tube-like structure. It's just like digging tunnel. If you have seen how tunnels are made for developing roads or rail tracks, you start digging tunnel from two different sides, and then you meet in the middle. Here, it's much more precise, much more engineered. Yeah, we already talked about germ band extension, so I think there's no need to go in further details. It's I would just like to again re-emphasize that this, because you must be wondering how most posterior part of the embryo, how can it move all the way up to here? This is, you have to keep in mind, this is due to reorganization of cells here. So you have cells like this. There's no increase in size of the cells or shape of the cells. It's just that the cells which are here on top, they fall in the middle here. And these two cells lose their connection. This one comes here, and this pushes the pole plasm cells all the way here, and then again come back. I take a break here, and after these morphological changes or anatomical changes, now we will look at the cell and the molecular biological changes which takes place in the pattern formation to set up the body axis after 10 minutes. Okay. If there are any questions, you can ask.